So let's talk about the experimental design. What is the purpose of blocking in an experiment? Okay. It's to help us to control for local variation or environmental variation. I find it's useful to know my research sites in the sense that I visit them at different points of the year, visit them after a heavy rain, and observe where does water settle in the field. What are the drainage patterns? What part of the field dries out sooner than others? Okay, so you can get to know your research locations without high technology approaches. Just use your observational skills and pay attention. And use that information to help you lay out blocks in your fields. So now I'm going to shift gear and talk about the data structure. So I want to just mention briefly compact versus flat formats of data. And I want to emphasize that you should be collecting your data in a format that is essentially ready for analysis. Okay? In order to do that, you have to know the statistical model that you're going to use before you collect the data. Okay? And this is part of good experimental protocol. All right? So, here's an example of compact format. Okay? So I have my replicates, which are stands, and I have my varieties or germplasm lines or accessions. Okay? And then I have some data collected for those. Okay? Compact format. All right? Here's an example of a flat format. Same data, but now I have variety in a single column, my block effect in a single column, and my data. Okay, and it's a percentage stand. Okay, so of the seed I planted, how many have survived? Okay, so these are the data that we're going to be dealing with. Our statistical model is going to be written out as a linear equation. Okay, and you should recognize this first equation as a simple linear model. Y is equal to mx plus b. Right? We're using linear models to perform our analysis of variance. And so my goal is to have you all able to articulate your experiment in the form of a linear equation. Okay? So this model tests whether there are differences between varieties with respect to the specified variable, in this case y, which could be yield or stand count. Okay? So the data that I just showed you actually has a rep effect in it, or a block effect. So we have to add a term. So our model looks like this. Y is equal to the average plus the effect of variety plus the effect of block plus error. Okay? So notice that for every main effect in our model, we have a column for the data. Okay? So most computer programs that you're going to use for statistical analysis are going to need your observations in a single column. Okay? You might as well collect your data in this form, which requires that you know the statistical model prior to collecting data. All right? So good experimental protocol means that you first articulate your model, set up the experiment that way, collect your data that way, and then you're ready to analyze it. Okay, so we're going to now talk about the bean root rot resistance screen. Okay, so this screen was set up for Rhizoctonia solani. There were strains from two different anastomosis groups that were used. So basically what's done is that grain is infected and colonized, and then that grain is used to seed a field that the beans are planted into. Okay? So again, we can look at a field like this. So we have this field day visit. So the field is actually set up with replicates in it. Okay? So these are blocks, and then the different accessions are actually randomized within each of these blocks. Okay? So if we're going to look at our data, so the data that was provided to you is in this file called 2year.rhizoc.csv. So year and rep are experimental design factors. So there are 2 years worth of data, and there are 3 to 4 replicates each year. Okay, and just to emphasize, in this field there are 1, 2, 3 replicates shown. Okay, so genotype and inoculation are treatments. So genotype is the varieties or accessions that were used. So those are all coded. Inoculation is either untreated or Rhizoctonia inoculated. So our data actually has this inoculated column that includes untreated rows as well as infected rows. And that's important. It's going to require that we make some decisions about the analysis that we actually want to do. And then the data that are collected are stand counts. Okay? And so that's a percentage of stand that is alive, and that was counted in June, July, and August. Okay, so we have, this is going to be Y, and we have 3 different Y variables. Okay, and so missing data in R is coded as NA. And that's important. You cannot leave a cell blank. You don't use a period like you do in SAS, it's NA.